Excellent. I see it's recording, so I'll go ahead and kick things off. Hello, everyone, and welcome. We're so glad that you could join us for today's webinar, Current State of Digitization in Mammalogy and Future Directions. My name is Shyla Davison, and I'm the Education and Outreach Coordinator for iDigBio. And before we begin, I'd just like to take a few minutes to talk about iDigBio and the work we still have going on. So as many of you know, iDigBio will officially conclude very soon. And for the past 15 years, iDigBio has had an incredible impact, supporting research, collaboration, and innovation on a national scale. This isn't a moment of sadness, but one of transition. And as this landscape evolves, the NSF does continue to fund biodiversity, digitization, and related efforts across the community, building on the foundation that programs like iDigBio has helped establish. And we're excited to continue these conversations and highlight the work happening across the field. So we're proud to pass the torch to the broader community, confident that the mission of advancing biodiversity, digitization, and discovery will live on in capable hands. We also wanted to share a brief update regarding iDigBio's timeline and its online resources. So while iDigBio is in its final year of NSF funding, we have been approved for a no-cost extension, which will allow us to continue into late 2026 or early 2027 as we close out the project. And as we continue through this period of change, I want to take a moment to share important updates about how you can still access iDigBio resources. Our website and wiki have now been fully archived, ensuring that the full record of iDigBio's work over the past 15 years remains preserved and accessible to the community. At the same time, iDigBio's long-term home has transitioned to a new GBIF-hosted portal where users can continue to search biodiversity data, including all data aggregated by iDigBio, and stay up to date on all ongoing news and activity. The current iDigBio website has been replaced with a simplified landing page that links to key resources, including the archive, the new GBIF portal, the Zoom Events Hub, and our legacy data portal. And the legacy data portal will remain available for searching through August 31st, 2026. However, data ingestion will conclude May 1st of 2026. So also related infrastructure such as the IPT has now migrated to GBIF. These updates are all part of ensuring continued access, stability, and long-term support for the data and resources this community relies on. So let me give a quick overview of who we are and what our mission has been, and the exciting work still happening. So iDigBio began 15 years ago with the support of the National Science Foundation and the vision of its founding leaders. Over that time, we've made tremendous strides together as a community, and our shared efforts have helped shape the future of biodiversity research. And as its heart, iDigBio has worked to make biodiversity specimen data widely available and useful through digitization, training, open access, and creative new applications. And while iDigBio as a project will eventually close, our mission and the data the community has generated will live on. And thanks to contributions from more than 1,000 biodiversity collections across 350 institutions in the US, the iDigBio portal now hosts nearly 150 million specimen records and 67 million media files from about 2,000 datasets. These data will remain available long after iDigBio, continuing to serve researchers, educators, students, and the public. And we're not done yet. As we move towards the conclusion of the project, we still have new initiatives on the horizon, such as this webinar series, and of course, the upcoming Digital Data Conference. These initiatives highlight protocols, practices, and strategies to ensure long-term continuation of collections digitization and data mobilization. The collections community is well positioned to sustain this important work, and we're happy to support that transition. And as we look ahead to the future of biodiversity data, today's webinar takes a closer look at how mammal collections have evolved from early digitization efforts to the advanced data and imaging approaches we see today. It also considers what comes next as new technologies continue to expand what's possible. So before I hand things over, I just wanted to mention that the chat and mic functions will be disabled today. So please post your questions in the Q&A feature, and we'll get to as many as time allows at the end. So thank you for your understanding, and I'll stop sharing my screen. Looks great.
Thank you, Shyla. 1 second while I navigate. Okay. Well, thank you everybody for joining us today and for iDigBio for hosting the mammal community in this effort to sort of spread the wealth of the work that they've done for the past little over a decade and hope that we're as a group not just in the mammal community but elsewhere within the Natural History Museum community able to sort of take that torch and keep it going as long as we can. So my name's Cody Thompson. I'm the Mammal Collection Manager and Associate Research Scientist at the University of Michigan Museum of Zoology in the Department of Ecology and Evolutionary Biology. And I'll start off today by giving the group here an introduction, but as well as orient everybody on mammal collections, where digitization has been, and where we're hopefully going to go in the future. So just a brief outline. I'm going to start off with giving a little bit of a background on natural history collections. For this audience, this will be more of a review. I'll talk briefly about collection digitization and provide a timeline as to where mammalogy has been and where we started at and where we are currently. And then I'll move into the format of the webinar and introduce the rest of the speakers for today. So natural history collections, a brief history. Again, this audience is obviously very well versed in natural history collections, but just to make sure we're all on the same page of understanding as to where the mammal community is sort of starting at. Really, I think all of us, to some extent, start our collection started sort of with the uh, European exploration, and I think it's important for us to recognize that a lot of the efforts within colonial times, um, they weren't always sort of grounded in what we do today, but that's certainly where a lot of our collections began. And sort of these ideas of cabinets of curiosity were really the foundations of much of what we have today in the natural history collection world. That's sort of the change, however, with Linnaeus and others, but in particular Linnaeus and uh, in his efforts to sort of standardize what we understand as biodiversity, and providing sort of this sort of standard way of viewing species, and providing the binomial nomenclature for uh, creating what we still use today as our sort of our baseline for systematics and taxonomy, and has been codified in the zoological code and other similar codes. Um, of course, that began to shift in the early, or excuse me, the mid 1800s with the work of Charles Darwin and Alfred Russel Wallace, sort of uh, sort of taking what was more of a very descriptive understanding of biodiversity and sort of switching those efforts to looking at the relationships of organisms to one another. And of course, Darwin's famous book on the origin of species provides much of the foundations for that we're still using today. And this really kind of led natural history collections to sort of shifting to more of a scientific endeavor that we again now see today in the U.S. And then with mammalogy and our colleagues in ornithology, that really those collections really began to shift even more so with the establishment of what was used to be called the U.S. Division of Economic Ornithology and Mammalogy. This agency, government agency, formed the baseline of what is now known as the Smithsonian's collections. And for much of mammalogy, that effort was led by C. Hart Merriam, somebody that um, uh, those of us in the mammal community regard as sort of one of the foundational figures in our science. Um, and we even recognize our top researcher annually with the C. Hart Merriam Award. So, but really, again, moving collections in a direction that we see today. But it wasn't until uh, Joseph Grinnell that sort of our understanding of how collections are utilized and more of sort of foreshadowing of what they could be utilized in the future was really established. And I noticed there's some folks from Berkeley here. Grinnell was a faculty member in Berkeley and really sort of developed out those sort of foundational understanding of getting data um, into a way that was reusable, right? And not only that, sort of setting the standards of how we record data in museum collections. And for mammalogy, much of what Grinnell set up and his time is very much still utilized today in how we record some of the basic information about the organisms that we study. One thing that's with Grinnell that was really is profound now looking back over 100 years ago is that he was very prophetic in terms of where he thought museums and museum collections can go. And this quote here from Grinnell that points out that at this point, I wish to emphasize what I believe will ultimately prove to be the greatest value of our museum. This value will not, however, be realized until the lapse of many years, possibly a century, assuming that our material is safely preserved and this is the student of the future will have access to the original record of faunal conditions. So Grinnell, really took those collections that were being established by these earlier individuals and really kind of set the tone for what we do today and how digitization looks in the collections that we curate and manage. Okay, so now shifting gears to collection digitization and mammalogy specifically. So historically in the mammal collection community, what we think about as digitization often starts and ends at sort of the computerization of our paper catalog. So those records that we're writing in when we accession and catalog material into our collections. This, of course, may include things like georeferencing, and this is sort of varied over time. And of course, as we do our work now, that's a critical piece of how we enter our collections into the public good. But it also might include things like imaging specimens or our paper collections or archiving media specimens. But the mammal community, and as we'll see here in a second, uh, has become expanding our sort of our breadth of what digitization means, and but it does kind of providing a little sort of a reflection at this juncture as to where we are in our digitization efforts. So I like to use the UMZ Division of Mammals as an example to sort of establish a timeline as to where we are today in digitization. So, looking at the UMZ Mammals collection, our collection is relatively old. Our university was founded in 1817, so we just surpassed the 200-year mark a couple of years ago. Um, our formal collections were established in 1837 as the Cabinet of Natural History that came when Michigan became a state. The museum that I'm in, the University of Michigan Museum of Zoology, or the UMZ, was formed as an independent research museum in 1913. And in 1977, with the funding support from NSF, the UMZ Mammal Collection began to digitize our paper records. So almost 150 years went by from the moment that collections started to assemble with the University of Michigan to the point where those collections, at least in mammalogy, started to become computerized or database. And here kind of showing two images of individuals that were really critical in that history. Lee Dice, who was a curator of mammals in the early part of the 20th century, and Phil Myers, who recently retired here about a decade ago, actually doing some of the initial data entry of our catalogs into a computer. So moving on, so yeah, again, we covered about 150-year history. Moving on, the UMMC collections starting in the 80s, we achieved our full computerization by 1982. And I recognizing that mammal diversity is much less than most organisms or other organisms. That's a 40-year head start from some of our colleagues, say in botany or entomology, have had. Um, or 30 or 40-year head start. And of course, much of, again, the other museum collections follow this similar timeline. By 1984, the UMMC mammal collection began digitizing our special collections. By 1994, the UMZ mammals began using FileMaker and supporting our own databases internally on our own PCs rather than using the university mainframe. So this Victor computer, which was uncovered in a closet during our move, was one of those computers that functioned as one of our initial computers for our database. Um, and then sort of moving on, just as a kind of a comparison of the timeline, 1994 also was the date that the Animal Diversity Web, which many of you know, was established by Phil Myers within our museum. Moving on to the 2000s, things start speeding up, and that sort of digitization takes a new a shift in mammalogy, and uh, in 2001, 17 mammal collections, including the UMZ mammals, created the Mammal Networked Information System, or MANIS. Those of us that were in graduate school or studying at the time are very familiar with MANIS, and it was a sort of a great initiative. And here's one of our students pictured here that was part of those digitization efforts that led to the georeferencing that Manus provided as well. 2001, GBIF was established. 2002, we did our first imaging project in the UMMC Mammals of the Great Lakes Flora and Fauna Project, which was funded by the Institute of Museum and Library Sciences. In 2007, the UMMC Mammals was fully georeferenced, excuse me, via the Manus project. So again, kind of looking at that timeline, 150 years had passed in our collection before we were computerized, databased. Another roughly 25 years, actually 30 years, excuse me, passed before we were fully georeferenced as a collection. Okay, moving into the 2010s, VertNet was established with NSF funding. At the same time, the Advanced Digitization of Biodiversity Collections program was created by NSF, which led to iDigBio and many of the TCNs and PEN projects that we are all very knowledgeable of. Uh, 2011, iDigBio was created, as Shyla just mentioned. Um, and then by 2013, Manus had gone offline. However, it was replaced pretty quickly by another portal, VertNet, which is still active today and has kind of been reinvigorated itself here recently, which we'll maybe get to a little bit later. Um, in 2014, our portals, our data were all available on GBIF and iDigBio. And then again, moving a little bit quicker here through 2015, the University of Michigan, we joined the Specify Consortium. And this is similar to Arctos and other large consortiums. We work with Kansas and Florida and other institutions to help the community. 2015, the Animal Diversity Web no longer supported. 2015, VertNet no longer supported, at least by NSF. And then 2017, so going back to that timeline, so 200 years after the establishment of the first sort of collections of the University of Michigan, the first true digitization project in the sort of the imaging sense was established that included mammals. Uh, More recently, 2018, the FuncQEE award was awarded, and we'll talk about that again a little bit later, which focused on rodents. Um, the BatPEN grant, which focused on phylostoma bats, was awarded in 2020. Ranges TCN, which and these are projects that are all mammals, was awarded in the same year, 2020. Um, and here we are in 2026. So, as of right now, according to GBIF, there's just shy of 7 million vouchered mammal records available. So, again, much smaller than maybe some of our peers and other collections, but um, a lot of those are they're pretty current. Those data are pretty current. Though what's lagging, of course, is the phenotypic data that many of the other collections have uh, developed. So that's what the this looks like, right? I mean, we have a lot of collections, a lot of areas that are still understudied within the mammal community, but we're heading to a point where we're really well in a good position for moving into the next phase. Okay, so what's next for mammalogy? Well, I'm gonna let my colleagues talk about a lot of this. The first speaker will be Charlene Santana. Charlene is a professor and curator at the University of Washington. She's been an active member of oVert TCN, or as a co-PI of oVert TCN, Ranges TCN. Her topic will be focused on the CT scanning efforts within the mammal community. And just a real quick fun fact about Charlene, she has her own Wikipedia page. So um, I, I feel great introducing Charlene in that manner. Second will be Brian McLean. Brian is a newly minted associate professor at the University of North Carolina, Greensboro. He was a co-PI in the FuncQEE and the PI in Ranges TCN. He'll be talking about trait data in the mammal collections community. And just a fun fact about Brian, Brian still thinks Appalachian State can beat Michigan. Nate Upham will be following Brian, and Nate's an assistant professor at Arizona State University and a co-PI in the Ranges TCN. And one thing I forgot to include here, he's been a leader in the mammal diversity database. Nate will talk about translating specimen data into different taxonomic perspectives. And just a fun fact about Nate, he is a pro at mammal phylogeny t-shirt design. Emily Breaker at the University of Colorado Museum of Natural History will follow Nate. Emily is a collection manager at the vertebrate zoology, was a co-PI and oVert TCN in the Omesa pin, which is focused on snakes. And Emily's topic will be focusing on digital data and extending the specimen paradigm in mammals. Just a fun fact about Emily. Emily is the sort of a Swiss Army knife of collection managers. She can do it all, and is a fantastic birder. And then to close out our group, Noe de Sancho who's an associate professor at DePaul University, and a co-PI in the FuncQEE will present on what we could do if we had the data, additional data, new data. And Noe, as you guys will probably see, is probably giving his talk on a beach somewhere. So with that, I thank everybody for joining us, and I look forward to the talks of my colleagues. So with that, Charlene, I'll let you take over.
Perfect. Thank you, Cody. That was a really, really nice background, and thank you for the introduction. So as Cody mentioned, I'm going to be talking about how we have been advancing mammalian research to CT scanning. But first, I will tell you a little bit more about the work that we do in my lab, which will contextualize some of the things that I'm going to talk about today. So In my lab, we're interested in understanding the ecology, evolution, and morphology of mammals. We integrate methods such as those within the fields of morphometrics, biomechanics. We do this in a comparative context using phylogenetic comparative methods, and we also look at the behavior and the ecology of organisms. And we're interested in understanding questions such as what are the mechanisms that underlie the diversification of mammal clades, and very often those mechanisms involve the evolution of novel morphologies or novel behaviors. And so we're also interested in understanding how these evolved phenotypes, so morphologies and behaviors, allow animals to use ecological resources. And as you might have guessed from the picture here of this very cute bat flying in the slide, most of the work that we do focuses on bats because they're an incredibly diverse group of mammals, but we also work on other mammal groups. So as my lab approaches these questions and we think about quantifying the morphologies of bats and other mammals, we face a dilemma that many other researchers in the area of mammal morphology and evolution also face, which is which methods do we use to quantify these morphologies? Because on one hand, we have traditional tools that such as dissecting the specimen if we want to look at their muscles. We have calipers to take linear measurements. We can take photographs and take measurements from those. And I'm calling these the 2D traditional methods, which tend to be much cheaper because of the equipment that it's involved in quantifying data in this way is very cheap. This allows for faster data collection. That means that we can measure many specimens, but it also means that the data might be coarser or less precise, especially if we're trying to measure small features. And also, if we're thinking about quantifying the morphology of soft tissues like muscles, it has to be destructive because we have to dissect out those tissues. And then on the other hand, we have newer 3D methods such as CT scanning, which is basically taking X-ray photographs of the specimen and then reconstructing the morphology in 3 dimensions in a computer. And these methods tend to be more expensive, first because of the barriers of acquiring the equipment. Sometimes there are costs associated with CT scanning, which kind of slows down the collection of data and means that we can include fewer specimens in general with these methods. Also, the processing of the CT scans is still can be a very time-consuming process because the human eye still is the best tool to tell apart individual features when we're looking at a CT scan. But we can get much more detailed data. We can get data in 3 dimensions. And because we are using X-rays and it's not destructive, we can look inside a specimen without having to cut it apart. So to overcome some of these challenges that these type of 3D data, such as CT scanning, brings up, there have been a lot of initiatives, especially in the last decade, and as Cody already alluded to, that have allowed for CT data to grow basically at an exponential rate and make it available to the scientific community. And of course, one great example of that is the oVert project, which generated CT scans for thousands of specimens across all of vertebrates. And as Cody mentioned, there are also some other initiatives funded by NSF that have expanded these data sets into more specific taxonomic groups, including the BatPEN, which is generating around 1,000 CT scans for 240 species of bats with a really dense geographic and ontogenetic coverage. Also, Cody, who is a PI on the FuncQEE, the digitization pen, is also expanding the digitization to CT scanning of another incredibly diverse group of mammals, that is rodents, generating 1,700 scans for 1,700 rodent species. And ranges, of which one of our speakers, Brian McLean, is the lead PI, is also providing resources for especially junior scientists to also generate CT scan data and other types of 3D data through many awards that are focused on generating more interest-specific coverage within the different taxa. So all these efforts, as well as the efforts of many labs who are also using CT scanning to generate data for their own purposes, are providing the whole community with a really incredible breadth of information. And I just searched this week to see how many entries there were on morphosaurs for mammals, and this includes CT scans, but also perhaps laser scans, and there are over 46,000 entries, which is incredible when you think about the amount of time and effort and resources that has to go into generating just one CT scan. Now, having the data is only one part of it. Also, we need the tools to process those CT data. And these tools have become much more accessible. And I would say that right now they're more accessible than they've ever been. So there are especially free platforms such as 3D Slicer, which is an imaging computing platform where you can process your scans, you can collect data. And the nice thing about it is that it's very well documented. There are trainings that are online, and the community is developing more and more tools. And I should say that in the past, we had to rely on biomedical software that is often very expensive. And so as part of the developments within this platform, we have packages such as SlicerMorph, which can be used to visualize, segment, digitize, and analyze data, as well as to share it. And then on the other hand, once we have our 3D models generated, there are also many packages such as Geomorph in R that we can use to then quantify the morphologies that we extract from the CT scans. So these tools are, again, opening the door for the use of CT scans for the study of mammals to a really wide community of users. And I say from personal experience, even in my classes that I teach, mammalogy and functional morphology, my students can learn to use these tools in just a couple of months. So the accessibility to the CT data and the ability to extract data from the CT data is just really, really impressive these days. And I would say also, in addition to all this data accessibility, the availability of CT scanning data has also enhanced the types of questions that we can ask in the study of mammals. And I would say one place where CT scanning really shines is in the research of small mammals and structures that are, of course, internal or complex. And I know this from experience because I study animals that are very small, such as bats, and I'm interested in parts of the mammal that are very complex, such as the skull. So instead of trying to take measurements with calipers or figuring out some really way complicated way to quantify these morphologies, we can CT scan these animals, and then we can answer all sorts of questions in the areas of anatomy and morphometrics or biomechanics. So here's some examples of the types of things that my lab and many other labs use CT scans for. We can use them to look at internal bony anatomy, soft tissue anatomy. We can do very complicated shape analysis. And in the area of biomechanics, we can do different types of modeling without having to even touch the specimen. For example, the figure that you see here in the lower right corner, we can do finite element models in which we are testing how different forces act on a structure, in this case, a skull, without having to do physical testing on the structure or potentially breaking it. So in the rest of the talk, I'm going to give you some specific examples within these broad areas of anatomy and morphometrics and biomechanics and biophysics. And because of my bias, they're going to be mostly about bats, but please know that these areas have been explored in many other groups of mammals. So first, I wanted to show you a little bit about the utility of looking at internal structures via CT scanning. And my work focuses on feeding, so I look mostly at mammal heads. So here's my bias. But one structure that has become a really interesting area of research that is not involved in feeding, but it's also still found in the head, are the terminal bones. So these are little scrolling structures that are found in the noses of all mammals. So you can see in this section of this mouse head. And the ones that are towards the front of the snout are functioning in conditioning the air. So they moisten and they warm the air. And the ones that are towards the back of the nasal cavity are involved in olfaction. And these structures, because they are hidden inside the skull, the only way that we had to study them in the past was by sectioning a skull, as you can see here with this deer skull from our collection. And even by doing so, it was still extremely difficult to figure out a way of how to quantify the morphology of these structures and then compare it across species. But with CT scanning, we can look at these structures without, again, damaging the specimen. And we can label all the different terminal bones. We can compare them across species. And we can do all sorts of quantitative analysis with them. So here's just a small sample of the diversity of terminal bones in bats that we have been studying. The ones in green are the respiratory terminals. The ones in yellow are the olfactory terminals. And we have created some metrics to quantify the complexity and compare it across species. And we're not the only lab who is doing this. These structures have been called the last frontier in mammal anatomy. That seems a little bit of an exaggeration, but certainly they are a very important structure for mammals that has not been studied in detail because we didn't have these tools before. And so other labs have also been looking at them, even at the scale of all mammals. And through these studies, they have been able to even overturn paradigms of what we thought these structures were doing. So there's still much more to do with these type of internal structures, of course, not only in the skulls of mammals, but all over their bodies. Now with CT scanning, the examples that I've been showing you have been focused on bony anatomy, and that is because the minerals in bones are radio opaque, so that's why we can image them through X-rays. That is not the case with soft tissues. The X-rays just blow right through them. But the community of sort of imaging technique development has come up with a workaround this challenge, which is to stain those muscles to make them radio opaque so we can visualize them through CT scanning. So several years ago, there was this very influential paper that took an old technique of iodine staining to stain specimens, vertebrate specimens, and image them through micro CT scanning. And the nice thing is that iodine binds to carbohydrates and fats in the tissues. And you can, through this method, you can visualize the muscles and the nerves in a structure. So you can see this rattlesnake head just with really beautiful detail. You can see all the muscles and ligaments and a lot of other things through this staining technique. And this can be done with basically any specimen from embryos to a large specimen. It all depends on the time that it takes to stain. So just to give you some examples of how we've been doing this in my lab and the utility, again, especially in small animals, we have been using these to look at the morphology of the jaw muscles across bats. And we can reconstruct them in 3 dimensions, look at the differences across the different species, and we can look at the internal anatomy of these muscles. For example, how the fibers are oriented, what subdivisions the muscle have, again, without having to touch or dissect these muscles or damage the specimen. This is a very time-consuming process to segment the muscles. Again, the human eye is still the best tool to do this so far, but there are new techniques involving computer vision and other tools that are automating these processes such we can automate and do measurements in these muscles that we couldn't do even with dissections, such as tracing the orientation of the muscle fibers. So again, a lot of opportunities here for more development of tools and generating new data. Okay, the second case study has to do with how we can use then these morphological data for biomechanical modeling. And I'm going to give you an example of how we model the biting forces of bats and how CT scanning can help us do that perhaps better. Just as a very quick background, this study was based on phylostomid bats, so the Neotropical leaf-nosed bats. They're a really cool family of bats because they include species with diets that range from insectivory to frugivory, nectarivory, and even vampire bats. So a lot of morphological diversity and a great natural experiment to look at how good these techniques can be. And so the question that we had is whether or not these 3D representations really enhance our ability to predict feeding performance. Again, because it takes a lot of time to generate the CT data sometimes. And one thing you need to know before I show you some results is how we generated these data. So to calculate by force, we use a simple lever model. And all you need to know is that by force is predicted by the cross-sectional area of the muscle, and I'll tell you in a minute how that's calculated, and then the torque that that muscle produces about the jaw joint. And we can calculate that by doing different measurements of where the muscles are inserted in the skull. So traditionally, to do the PCSA calculation, we would have to dissect out the muscle to measure its mass, and then measure its different muscle fibers. We have to separate them and then also measure the fiber orientation. But with CT scans, we can reconstruct the muscle volume instead, divide that by the fiber length, which we can measure from existing dissection data or by the CT scan itself, and then we can calculate that. And so we did 3 comparisons. We compared sort of purely traditional approach using dissection data for the muscles and then taking those measurements from the skull to do the different levers of the muscles. And we call that this 2D. And we compare that with a hybrid method where we had dissection data from for the muscles and 3D skull data for the levers of the muscles. And then finally a fully 3D model where we had the PCSA calculated with the 3D muscle volumes and 3D skull data. And to make a long story short, and we compare the outputs of all these models with data on bite forces that we measure in the fields. And so what we got was that the 3D methods are actually more precise, so they tend to give more consistent results. And the fully 3D method tends to be more accurate, so the predictions tend to be closer to the bite forces that we measure in the field. The downside is that it takes a really long time still to segment out those muscles. And again, with opportunities to automate those processes, then maybe we can get to a place where we can fully automate and make these fully 3D methods be the most efficient, both in terms of the time that it takes to apply them, but also in the outputs that we're getting. So if you're interested in learning a little bit more of these comparisons on how the 2D approaches can compare to the 3D approaches, we published a paper several years ago in Integrative Comparative Biology. And still, I think the things that we said in that paper still hold today. And in addition to comparing the methods that I showed you for the bite force predictions, we also compare methods relating the 2D and 3D approaches when it comes to understanding the evolution of mammal skull shape. So to wrap up and to summarize, as I showed you, CT scanning and all the associated tools have revolutionized and reinvigorated the study of mammal anatomy. It feels like it's really cool now to study mammal anatomy again with these new tools, as well as functional morphology. And it has led to discoveries and new research agendas. And there are, of course, many opportunities still for expansion and automation of data collection and analytical pipelines to then sort of address those steps that still slow us down during this era of big anatomical data. And with that, I will thank you all for your attention, and I'll stop sharing my screen.
Thank you, Charlene. Okay, next will be Brian McLean. So Brian, if you want to.
How does that look? Someone tell me what you
All good.
see.
All good, Brian. Well,
Okay, great.
actually, click
Do-
your slide. I think it's there we go.
Good
Not
or no?
quite.
No. Just a sec here. Let me try again. Now,
It's just,
what do you see? Like the
I see
PowerPoint
a
window?
black screen.
Okay, yeah, that's not good.
Is that same thing for you, Lauren?
Yes.
And it doesn't look like we're in like the presenter mode either.
Yeah, how do I get in presenter mode? Let me try one more thing here.
So when you go put it into presenter mode in the bottom right, you can click the little like slideshow, and then when you go
Yep.
to share, there should be different options so you're not like just sharing from PowerPoint, but you can
Yeah.
share that shared screen.
Yeah, actually works. Sorry, everyone.
It's okay. We have time.
Still, yeah, how do I?
Hit the presenter view button. Try that.
Yeah, it's not working for some reason.
Well, if you want, you could share from here and just toggle through your
Mhm.
slides if that's easy.
Let me try one thing. I might have my camera off, but
Okay.
let me
Okay.
try this thing fast.
And folks, if you have any questions, feel free to drop them in the Q&A as we go, and we'll address those at the end of the session. It's just really small, Brian. I'm not sure.
Yeah. One more thing. You guys, I'm sorry.
Well, if you want to go back to your original, what you had, and just uh-oh. Did we lose Brian?
We did lose Brian.
Well, maybe while Brian tries to reboot or whatever, Nate, I don't know if you're ready to go. We can maybe move on to, yeah,
Yeah.
move on to Nate and then let Brian get logged back in and go. Nate, does that work for you? Awesome. Okay, so we're gonna come back to Brian after Nate's talk.
Cool. Yeah, glad to fill in. Let's see. And hopefully not have the same problem as Brian. How is that? Yeah. Okay. Thanks everyone. Yeah, so this is great. Yeah, I think and Brian's talk will make sense in this context too, because he's going to talk about digitization of range for the ranges project, which is specimen digitization across Western Hemisphere collections, in particular in North America. The idea here that we're going to be talking about is this idea of taxonomic translation, and I'll define that as we keep going. sort of to lay out the problem, something that I first encountered in building this large-scale mammal phylogeny for over 6,000 species from public data. And while this phylogeny enabled us to address some interesting questions, I think one of the main things that it was for me, and I think continues to be, is it really pointed out the gaps in our knowledge. So that while we were able to estimate things like tip speciation rate, there's a lot, like 25, it was actually 30% of mammal species had no DNA sequenced at that time. It's increased some since then. But at the same time, taxonomy continues to change really rapidly, and that's something we'll talk about more here today. And that's what we want, right? Like that's an increase in knowledge. But the real problem with it is that the ecological, genomic, and any data that's derived from specimens about species is largely unlinked to that taxonomic change. And so that really ends up being kind of the issue where we end up with a gradual decay in the accuracy of our occurrence data through time. So in a perfect world, all the biocollections data and all written knowledge would already be digitized and extracted and taxonomically updated and then ready to analyze. We'd just be like, let's go to that website and we can address any biodiversity related question that we have. And so it's not that case, right? That's what we're working at here. We have is a lot of what we're calling dark data that is kind of been written down, let's say, and some of it's been digitized, other has not. There's also just a large percentage of information that's in biological collections that's never been studied. But certainly biocollections are the basis for basically all written knowledge about organisms. And that what we can do is, rather than just having these digital data floating around in databases, we can connect them in meaningful ways. In terms of interactions like hosting interactions or roosting or food web type interactions, and the evidence of those interactions, like who was observing it, what type of measurement were they taking, was this a DNA sequence, so forth. So there's some ideas that kind of initially came together for us during the pandemic and thinking about mammal virus interactions and sort of our inability to really query back the way that we would want to about the reservoir hosts of various different viruses, and kind of got us thinking more generally about, if we want to address questions in phylogeny and ecology in a group like mammals that's supposed to be well understood, and well studied, that really what we need to be doing is linking those things through taxonomy, but that it's not like we want just one taxonomic perspective. This is a human construct that we're placing on top of organisms in the environment. And so we want the ability to actually shift between different taxonomies to treat it as a variable, so that we can say that my conclusion is robust to 3 different taxonomies, therefore I have a lot more confidence in it. Or conversely, like this conclusion that I have is, it only works based on the latest taxonomy or one from 2005. So we're not quite at the ability to do that kind of thing yet, but we're taking steps. And so today, be talking first about just what is our knowledge of species diversity in mammals, and how do we know that? And then sort of when does taxonomic change matter, and some approaches that we're proposing for doing this type of translation work. So thinking about mammal species through time, the 3 volumes of Mammal Species of the World are certainly kind of paramount in setting the stage here. There's some volumes before that too, but sort of, thinking about the full diversity of mammals in a modern context. This 2008 edition of the IUCN Red List is especially important because it was the first time that we had digitized geographic range maps for all mammals. And this continues to be kind of a resource that has subsequently been updated in these later versions of IUCN, kind of gradually increasing the number of recognized species. But it wasn't until the Mammal Diversity Database started, it was launched in 2018. We'll talk about that more in a second, but sort of this idea of trying to track the real-time changes in the literature for mammal taxonomy. And so this has been a really fun initiative that we got started. Connor Bergen and I kind of got this going initially through the American Society of Mammalogists. They've been really supportive. And most recently, Haru Hendika has helped us revamp the website. So there's a lot of neat things going on. If you haven't been there lately, go to mammaldiversity.org. There's even an app that you can download for offline use when you're in the field. But the kind of dramatic thing and the important thing for us here is thinking about the increase in species recognized, but not just increase, it's also kind of rearrangement of those taxonomies. A lot of new genera have been recognized. We now have a fully curated synonym list of over 50,000 names, getting closer to 60,000, to allow for some name-based taxonomic translation. Talk about that in a little bit. So this is all summarized in a paper that we had last year in Journal of Mammalogy. The sort of bigger, so that's like keeping track of change, but then there's all of the conceptual change that happens with that. And so in particular, when species get split, and it's really this question of, observations of species according to who. And so there's this broad scale taxonomic concept of a species, and then there might be a smaller one. And sort of the issue here is that the name of a species always goes with the type specimen. And so you might have a sensu lato version of a species that's fairly widespread and a sensu stricto of the same name. And that's not differentiable when you're just looking at names in databases. And so it's this sort of common problem where you have the same species name A is used with different taxonomic meanings. And so, I think we can all sort of agree on this, that these kinds of taxonomic splits happen, and that the digitization efforts that other folks will be talking about here today, in particular of traits and geocoordinates, can really actually help us update the taxonomies of those records. And, not just to the latest perspective, but perhaps to other intermediate perspectives that we might be interested in. Here's an example using some range maps, which ends up being a pretty nice way to conceive of a lot of these changes. So this is Miniopterus schrebersi and Fulginosus that we'll see are somewhat of a species complex in 2010 with these IUCN range maps. Some later range maps that have been digitized to the Mammal Diversity Database 1.2 taxonomy. Kind of really changed the way this looks, and it's sort of all overlapping, so I'll separate it here. But you see that the geographic concepts are quite different. There's been some species split out. There's some new names involved, but the old names are also still there, still valid. So when we kind of look more broadly across all bats and see, which bat species have been affected by taxonomic splits, it ends up being about 16% or 1 in 6 bat species globally have been affected by some taxonomic change since 2005. And that if we intersect that with viral interactions of those same species in databases, it's about 22% of all known bat virus interactions involve one of those species. And Miniopterus schrebersi is one of the top species. It's the top species for these ambiguous viral interactions. So that's of concern. And when we actually look at individual records in GenBank, we often see that the metadata that's associated with those occurrences is not sufficient to update the taxonomy. So something like this record, all we have is the location of China in 2012. And when we go back to the range map here, we see that China is no longer involved in any of these species that have been split out. So it's sort of, if there was a voucher, we could go back to it. Unfortunately, there's probably not in this case. So this is sort of like cautionary tale, and I think an idea that we need to be paying attention to these taxonomic changes and for the updating of records. And so there are these three-based approaches that we're proposing. The map-based one is what I'll talk about here today. But you can also imagine doing sort of DNA-based clustering if you had accurate reference database. Again, not always the case. And then name-based matching is just using a synonym list, which is what folks probably do most commonly, but has some issues with these splits. So sort of the extreme example that is a maximalist argument to say like, okay, let's look at this split of the most common North American mammal, Peromyscus maniculatus, that has many records on GBIF. And there was this proposal in 2019 to split maniculatus into at least 4 species, and sort of with some proposal about what those geographic ranges would look like. And so what we did, and because those geographic ranges are largely allopatric with respect to each other, we can literally just do polygon and point joins. And then for species that are sort of in the margins, so what we did here is the species that have white borders on them are kind of highlighted and needed to be resolved with additional machine learning from traits of those species. So we're additionally pulling in trait data in addition to the maps to kind of do this disambiguation exercise. But it's largely an inference using geospatial concept data, and then to some degree with traits as well. And so if you're curious about this, we actually have a GitHub repo set up to start doing some beta testing. Caleb Powell is a ASU PhD student who's been leading this effort. And I think, yeah, we're excited to start to share these tools with the community and have more people using them. And sort of, yeah, the name lore is this idea that, according to lore, we could still reuse old data, right? And, Cody was kind of alluding to that with this quote from Grinnell. But that, similarly, again, with viral interactions, this is something that people should care about with respect to what is the reservoir host species of something like C. nombre virus across North America. And we see that, it changes from being maniculatus to sonoriensis. And then there could be some important implications ecologically as to, what is that host doing? That that knowledge should be transferred to epidemiologists and so forth. So we had a paper about looking at this last year. But similarly, we're able to re-identify a lot of those interactions at the county level in this case, as extracted from papers. Just real quick, so in case some people don't know, we don't have to only use the IUCN range maps anymore. This paper in 2022 published some alternative geographic range maps from other sources, including Mammal Diversity Database 1.2. And so some folks in my lab, Angel and Norma, had been working on updating additionally from that 1.2 to additional versions. This is something that's ongoing. We've made some of these downscaled range maps available through an R package. So it's a little bit easier to work with the maps. They're not as heavy computationally. And we have these sort of 2 periods of change to potentially be working with in this context. So just to kind of wrap up here, we've got a tally of currently accepted mammal species. This is going to continue to change. We're going to want to continue to update and have the ability to reuse old data. So approaches like taxonomic translation seem to be important. And that we can potentially do this with range maps. We need to keep updating the range maps so that it becomes an additional thing that we need to curate and that the community ideally can do as a joint effort. But that, similarly, curating reference sequence databases and synonym lists is also important. And that, yeah, just this idea again of taxonomic translation and treating taxonomy as a variable, I think will be important for us. I want to thank a lot of folks that have been part of this work, in particular the BAT Eco Interactions Working Group that's been sort of the home base for developing these ideas, and our biodiversity group at Arizona State. And thanks. I can take questions at the end, it sounds like.
Perfect. Thank you, Nate. Yeah, and again, folks, as Nate mentioned, just pop those questions in the Q&A and we'll talk about them at the end. Brian, are you ready?
Yeah, I think I'm ready.
All right, go for it.
All right, does this look good? Great. Everybody hear me? So in some ways, we're gonna return to traits a little bit, but in some ways, going after Nate's talk makes a lot of sense because I'm going to dig down into basically the specimen level traits that are important for the next generation of doing mammalogy. And so traits are important because they interact with many aspects of an organism's environment. That includes things like morphology, life history, ontogenetic changes, ecological associations, and these are all interacting with other organisms and the environment to determine some pretty important outcomes like individual fitness, population persistence, and even at the longest time scale, evolutionary success of lineages. And I know what you're thinking. Mammals are a relatively well-studied clade, so trait data must already exist for mammals, right? And the answer is yes and no. So this is my attempt to put together a mostly complete summary of existing mammal trait databases that are out there. So I've listed a set of 5 that I'm most familiar with, going from Ernest et al. In 2003 to a more recent paper in 2021, the combined database. And these are databases of mammal traits. And the size of the circles are scaled kind of by the number of traits that are in there, and that would be the number of species in the database multiplied by the number of traits. And here I'm considering all traits in gray and then only morphological traits in yellow. So you can see the size of our trait database is increasing roughly through time. Morphological traits lag, but trait data are increasing through time. But the problem is that all of these databases compiled over the past 20 some odd years depend on the same foundation. There's not many new data in these databases. They all depend on the same foundation. That's mostly derived from existing literature and imputation. And this is a problem because we're not expanding our knowledge of intraspecific variation, like Nate talked about. We're not able to easily track taxonomy and the provenance of the trait measurements through time and through databases very easily. And then, one of the biggest problems is that these data exist at the species level. So we can't know much about variation within species. We want to know more about individual and population level traits because they're critical for monitoring responses to past and present global change. With enough traits from within a species, we can know something important about their phenology, their morphological characteristics and how that interacts with the environment, their reproduction, their physiology, their movement, all of which are things that we expect to change relatively rapidly under different global change scenarios. This is an example of where, from my lab, from where some individual or intraspecific level traits might be important. This is a study we did on litter size. One of my students, Amanda Weller, conducted in 2023. Litter size, specifically embryo counts, the reproductive output per reproductive event for about 43 small mammals. And what I want you to look at is the plot on the right showing, across all mammals, litter size decreases with body size. But within a species, those are the colored lines, the trends can be very different and they depend on which group you're looking at, whether the slope is positive or more flat. So we're really at the forefront of being able to have these data and use them for studies like this. Digitized collections will play the biggest role in filling the trait data gaps that exist. It won't be more data necessarily and traits from the literature. It'll be mining collections for these data. And just to make the point that AI alone won't save us in these cases, right? In fact, it'll make voucher materials and provenance more important. We need to know where individual trait measurements, the more and more we digitize, we need to know where they come from, which host and animal and individual they're from. That can be evidenced by this image I created on using ChatGPT, where the names of the mammals are not quite right. So, right, we need to track our new inferences back to old data and provenance. The reason why collections will be really important for extending our understanding of mammal traits is that standard morphological measurements are really well established for mammals. This shows a set of 4 measurements plus body mass, that would be 5, that are taken on a lot of mammals in the field and in museums. So these data are available to some extent in a lot of different forms, on tags, on labels, in catalogs, in databases, some of them. So there's a lot of data not digital that we need to harvest and use. The problem is that less than 10% of these of digital mammal records have detectable morphological or reproductive traits. So if you go to GBIF, any of the record sets, for example, that Nate talked about, 10% or less of them are going to have some morphological or reproductive trait data that can be kind of discerned and extracted from them. So this is a real problem. We just published a paper about this with the Ranges Network, which I'll talk about in a minute. This is just a plot showing the density of georeferenced mammal specimens for about 54 mammal collections. These are data from GBIF. When you look at the number of those that have detectable morphological traits, and this can be just one of some 10 or 15 different types of morphological traits, or reproductive traits, the numbers, the density of traits really drops off quickly. And you can see the distribution of those trait data from specimen records in North America. So the data are getting darker and harder to find and use because they're not there. So that brings me to introducing the Ranges TCN, which a lot of you have heard about. Some of you here are even involved in Ranges. This is a thematic collections network that's entering its 4th year now, as of tomorrow. And you can see the distribution of where collections are in the red triangles. You can see a list of people who are formerly PIs or collaborators with the network. We think in the network there's about 1.2 million mammal specimens from Western North America that are available to be kind of visited and visited for traits, essentially. We think that about 3 to 5 million morphological and reproductive traits might exist in this specimen set, but we don't know, and we won't know until the data are final. But Ranges is an attempt to basically do this, to harvest and digitize traits for all mammals from Western North America. Ranges really exists to change the fate of traits, to move them from analog formats or from digital formats where they can't really be detectable or they're very heterogeneous, and to standardize them and make them available for the community to use. So Ranges involves a lot of different things over the past 3 years. Digitization, either manually from the specimens themselves or from image transcription, development and augmenting of an existing trait ontology, extracting traits, if they're digital, standardizing them to names and units, and republishing them in a way that they're linked back to the mammal record. So our vision is really this, to build a more extended mammal specimen network where these different circles in blue, the morphological and life history and reproductive traits, can be kind of used for research. Ranges is publishing traits now. We've published a baseline data set of essentially pre-Ranges data, everything that existed in records prior to when we started working. And we're going to build on that and really quickly in the next month or so publish our combined year 1 and year 2 data of traits. So look for us on Zenodo, and we'll be announcing that at different places. And I just want to thank the Ranges leadership who are pictured here, but really the entire network who are doing the hard work of digitizing and supplying trait data. So far, 28 institutions and at least 55 individuals, and we're growing a little bit. So with that, I will stop since I use so much time trying to get Zoom to work, and I will just take questions in the panel.
Thanks a lot, Brian. Okay, up next is Emily Breaker. Emily?
All right, does that look okay?
There it goes. Yep, good to go.
Okay, great. Well, thank you so much. So I'll be talking today about managing extended mammal specimen data. Okay, so as we've heard from the previous talks, there's been many mammal-based digitization efforts over the past few decades, and at the same time, emergent technologies have allowed us to analyze our collections in new ways and invite different disciplines to engage with specimen-based research. And so together, these advancements have really generated large data volumes, complex information, and novel data types associated with our collections. And really integrating, linking, and presenting this information within collection management systems and packaging it for data aggregators are not trivial tasks. So for this talk, I plan to talk about some of the challenges and opportunities in managing extended specimen data that we face in the collections community, broadly speaking, as well as talk about some of the approaches that we have taken or that we're thinking about in Arctos. So zooming out, I wanted to first address a few of the major challenges to implementing and networking extended specimen data. And while these are largely technical, they are also social challenges since they do require community coordination, standards development, and implementation guidelines. So with data models, there's a lot of variation across systems in terms of data structure and standardization. There's a host of different collection management systems available, and certainly some collections that are still using spreadsheets to manage data. And in some cases, this translates to a limited ability to link out to extended specimen products like genetic sequences, CT scans, or publications that relate back to vouchers, or to connect related or co-collected specimens to one another. And at the aggregator level, there's also structural challenges. So as data providers, we're ultimately flattening our relational data to package our records to GBIF or iDigBio or VertNet. And we have complex data that don't always map cleanly to Darwin Core that become unstructured in the data publishing pipeline. And for many researchers, data aggregators are the main entry point for accessing mammal data rather than the source collection management systems. And so therefore visualizing extended data might be less dynamic than when viewing the canonical specimen records. Aggregator data models are also built around occurrence records versus collecting events, which tend to be very useful nodes for tracking data streams flowing from co-collected specimens like hosts and parasites, and it can make it difficult to illuminate those connections between specimens. So in essence, both aggregators and collection management systems are still very much catching up to the extended specimen concept and require more infrastructure for fully implementing and representing extended data. So another major technical hurdle is the uneven use of persistent identifiers. So these are essential to creating that constellation of interconnected data that's really at the heart of the extended specimen paradigm. And both the collections community and our partners really need to embrace the use of interoperable identifiers to fully automate the process of linking objects and communicating information across the semantic web. So these are some examples of persistent identifiers. And so right now, many of these connections, whether that's, specimen to dataset or specimen to publication, specimen to CT scan, are made manually. And so some of these connections are incomplete or non-reciprocal, or they're difficult to access or invisible to downstream users and even us as operators. So for instance, you might be totally unaware of sequence data that's sitting in NCBI that relate to your collections. Or if you're having to manually round trip data like georeferences or annotations or transcriptions versus this information sort of automatically flowing back into your source system, errors and inefficiencies are more likely, as well as lags in access to this improved information for end users. And connected to persistent IDs is the use of APIs. So much of our data is currently siloed, meaning that many key repositories as well as collection management systems lack APIs that would leverage PIDs to allow for dynamic linking and cross-repository querying. So building on this, something that we've been thinking about in Arctos in terms of persistent identifiers is that we need even finer resolution identifiers as we integrate more complex data. So most identifiers currently operate at the specimen or record level, but implementing identifiers at the preparation level or part level, as we say in Arctos, confers more granular precision. So for example, if we have an ABERT squirrel that's been prepared as a study skin, a skeleton, and 3 different tissue types, we want to be able to directly link the publication specifically to the skull that was measured or the GenBank accession number to the liver sample, not just to the parent record. So it's really clear what preparation type or sample was used in the study that's being cited. And so we're still figuring this out as a community. We're currently using part IDs, which we call unstable since we can technically delete parts and we don't have to, or we don't have redirects in place, but we may formalize these or adopt something more persistent and resolvable like ARCs in the future. And this kind of resolution is also important for other types of mammal data like serial samples from tagged or captive animals, where multiple samples are taken from the same individual at different times under different conditions. So really being able to trace resulting products to those specific events, sample types, and not just to the overarching occurrence record is really essential as we develop out this concept. And so despite these challenges, there are tools emerging to help manage and link extended specimen data. So I wanted to highlight a couple of Arctos examples. So this is the structure of a basic Arctos record. It's fairly straightforward to collate and display extended specimen products as embedded media or to link out to derivative data, as well as create linkages between Arctos records. And so all of the blue hypertext that you see indicates connected data, and the blue arrows are linked out to external resources. So in this example, I have a montane shrew from our collection. So the specimen has been cited into publications. So there are DOI links to those journal articles. A Hermite was found on this individual and prepared on a slide. So there's a hyperlink to easily navigate the parasite record, which of course shares all the same event information. The shrew was CT scanned, and there are Morphosaurus ARC link outs and media objects representing the volumetric data. And there's also a ledger scan hosted by the CU library server that can be viewed when clicking on the thumbnail. And so there's also automated links to the same record in aggregator portals. So you can see down at the bottom, GBIF, iDigBio, and Globi. And Arctos has tools to detect and track extended data. So the first are Morphosaurus bots. So these are a set of two tools that use the Morphosaurus API to search for Arctos GUIDs and retrieve related ARC identifiers to automatically create links and media objects in Arctos that navigate to the corresponding Morphosaurus record. So you can use these bots in lieu of manually bulk loading this information yourself, and they're particularly useful for detecting when a researcher or another institution has scanned loan material from your collection and directly uploaded it to Morphosaurus. So it's a really nice example of combining a stable identifier with API-based automation. The second tool is the GenBank Finder that monitors NCBI for unlinked specimens. So it searches on elements of the Darwin quadruplet to suggest sequence data likely belonging to Arctos collections. So I actually just clicked on this tool last week when I was screening the slide deck, and I found sequences related to 5 spotted cat specimens that were loaned back in 2014. And the GenBank information was just posted as well. So I had longer about this alone. So the service is dependent on researchers entering tissue source information into NCBI to begin with, which we all know is a big if, but they don't have to format it perfectly. And even though this is a semi-automated process and relies on verification by staff, it does serve as a model for connecting objects without resolvable identifiers. And Arctos also provides a daily snapshot of specimens using NCBI identifiers to the link out system, so that NCBI will reciprocally point back at our vouchers and allow users to easily navigate between the connected records. The third tool is the reciprocal relationships bot. So in this example, a parasitologist recently visited our collection and found 2 parasitic mites on a deer mouse specimen. I cataloged the mites and created parasite of relationships with the deer mouse host record and collected with relationships between the 2 mites to link these different vouchers. And then you can see that the bot then added host of relationships in the deer mouse record, pointing to both mite records, closing that data loop. And this would have been a separate step for me because I can't load host of relationships until the parasite records were created. So only identifying won't have that relationship between Arctos to the rest really ensures connected data won't fall through the cracks. And you can imagine that this sort of cross-linking can really scale, working with multiple samples from the same collecting event. So the bot is a real-time saving measure. There are many other relationship types that we can use to connect records. So these are sort of the most relevant to mammal data. Another challenge our community faces when carrying out digitization efforts is ingesting data. So whether transcribing labels and measurements from images, as with ranges, conducting a crowdsourcing project, or pulling in georeferences, there really needs to be a way to easily append this information to source records in a granular way and at scale in whatever collection management system that you use. So in Arctos, we have numerous component loaders that make it possible to modify and delete data using bulk upload workflows. But even with these tools, there are bottlenecks in preparing spreadsheets versus automating ingestion from external sources is labor intensive. And that's not to say that automation tools, including AI, will solve everything. Yes, they can absolutely streamline workflows, but I want to underscore that they don't eliminate the need for trained staff and territorial judgment. So achieving the extended specimen absolutely requires investment in both human and technical infrastructure. Holistic sampling builds on the extended specimen concept, where multiple specimens and samples are co-collected from a single event spanning multiple disciplines and potentially deposited across different collection types or even institutions and data repositories. So for this example, a chipmunk is collected with parasites, nesting material, a soil sample, environmental sensor data, which ultimately yields multiple vouchers, various sample and preparation types, media and analyses, all of which connect back to the original event and to each other. And so a major opportunity here is engaging with other disciplines that could benefit from working with holistic and extended data that might be unaware of or underutilizing our collections. And so part of this process really involves learning the languages and needs of these other fields. And so a small example that's quite useful is that that's quite useful for pathologists and parasitologists to know if navigate sound processing specimens so that they can calculate rates of infection. So therefore, getting negative findings is something very simple that collections are built into data and workflows that would meaningfully expand the utility of our data for these communities. So in Arctos, we've built out examined and detected fields with controlled values that can accommodate extended data by including pathogens, parasites, and contaminants. And this kind of targeted development is shaped by actual project and user needs in a way that, it's a way that collections can build bridges with other disciplines and foster collaboration across domains. And the goal is not only to provide structured, but research-grade data when possible. So this means creating community-defined terms with clear definitions or pointing to established authorities, as well as attributing every data point with an agent, a date, and a method when possible, so that it's really clear how a value was obtained and by whom. And these can be layered, so that skull measurements taken with calipers in 1994 can be distinguished from the same set of measurements obtained via CT analysis in 2026. And so I wanted to promote the idea that creating shared ontologies and controlled vocabularies across the mammal community would also make data more consistent and discoverable. Another opportunity in working with user communities is to really understand what they need from extended specimen data and design for it to increase access and reuse. So different research communities have different needs, and something as simple as building lightweight APIs that sit on top of our collection management systems or aggregator portals and serve as a translation layer could really offer simplified discipline-specific interfaces to streamline data discovery and really provide access to the data that's most relevant to these folks without a steep learning curve. So it'd really be great to have more conversations and curator buy-in to create more connectivity between the research and collections community. And such feedback loops are ways that collections can remain central to emerging research. And just finally, as we build out the extended specimen model, a discussion point we're thinking about is what information do we maintain within our own collection management system versus what information belongs in external repositories? So essentially, where do we draw the line between what we think of as collections versus research data? So in practice, most collections store primary collecting data and curatorial information in collection management systems, while derived products live in dedicated repositories that are purpose-built for handling those types of data and are well known to the research community. But sometimes the lines are blurry when it comes to things like trait data or media that's digitized by collections like CT scans that are then externally hosted. And there are also sustainability questions. So we're ultimately building an infrastructure that depends on the long-term stability of external repositories, and hopefully they will be around in 100 years. And that's very likely the case for the well-established sites that serve broad user communities. But long-term support is probably not going to be true of every repository out there. So it might be worth thinking about how we prepare for or provide continuity if a key repository sunsets. So I think it's useful to reflect more about collections' role in the extended data ecosystem. Are we finding aids and hubs for these increasingly diverse data streams, or should we also be collating everything known about our vouchers, at least in some sort of synoptic, snapshotty way? So just fruit for thought. But just to wrap up, connectivity and interoperability are essential to mobilizing extended data and in unlocking insights from our collections, especially as data richness and analytical tools continue to grow. But this framework does require investment in people, as well as technical infrastructure and in community coordination to harmonize our efforts. And we need more sustained conversations across our communities about extended data management needs and priorities as we continue to develop out and realize this idea. So thanks so much. And I think we'll take questions at the end.
Great. Thanks, Emily. Next will be Noe de la Sancha. There you are, Noe. Go ahead and share when you're ready.
Okay, you see me okay? Okie dokie.
All good.
Okay, I'll get started then. Muy buenas tardes, damas y caballeros. Thank you for having me. I genuinely appreciate it. This has been an awesome experience, and I'm kind of glad that I went at the end, given that a lot of my colleagues have kind of brought up a lot of the items that I was hoping to had to touch on, but this is a good follow-up as we think about the application of collections to more field-related research. So I am primarily a mammalologist and an ecologist. A lot of my research has dealt with the effects of habitat-driven habitat change on biodiversity, and I use small mammals as a proxy to understand these patterns. One of the central themes that has been helpful to understand these is the application of museum collections, and I will try to sort of touch on various aspects of this at multiple scales of ecological questions, from the population to the community level, to the ecosystem and landscape, and then more into aspects dealing with zoonotic diseases and perhaps bigger scale patterns. The next thing to consider is that at the forefront of ecology is this whole new emerging discipline, which is urban ecology, for which I will address is coming into its own right, and museum collections are going to be important for answering many of these questions moving forward. So as you may or may not know, we're currently in the sixth mass extinction. This has been one of the largest loss of biodiversity, primarily driven by human habitat loss, fragmentation, and degradation all around the world. One of the things to consider is that as collections, we're still at the very end of this, and many people have highlighted this idea that perhaps museum collections are having a big impact on natural populations. I would argue that these are minimal compared to what the true impacts of on biodiversity are currently, and museum collections are extremely valuable for future understanding of biodiversity-related questions. As we think about this, there's been this sort of like tension between taxonomic and old school evolutionary biologists and ecologists, whom have had this idea that collecting voucher specimens is impactful on museum collections. And I would argue that in most parts of the world, it is highly important that we keep monitoring and also collecting small mammal populations in order to better understand the bigger picture of what's going on our planet. As you all may know, field collections are complex. They are very labor intensive. They also add biases because there's only certain places on the planet that we've been able to sample. By no means do we have a complete inventory of the biodiversity of our planet. And many small mammals in particular are highly, highly underrepresented in collections. Biases include a lot of the logistics that are involved in getting to many of these places on the planet, and also making sure that these museums make it finally to museum collections where they're preserved, hopefully for long-term use. I'm not going to go into much of this, but it's fair to say that our collections around the world are vast and valuable. However, there are still many gaps from many taxonomic groups, and many of the aspects that I think are valuable moving forward for research in ecology are many of the items that my colleagues have pointed out, things like CT scans, linear measurements, pictures of specimens, pictures of skins that are also very valuable for things like a lot of the distribution maps and understanding of where and how these species are found on our planet. And also when we start thinking about not only many of the cranial specimens that we've been looking that are part of many CT scan analyses, but the postcranial samples and measurements that are going to be very valuable for other aspects of ecology that I'll get into in a second. Now, when we think about ecology, one of the fundamental things is being able to understand what species we have in any particular location. This might seem like a pretty straightforward task. However, doing field work for a long time, many species of mammals, especially things like rodents and bats, start to look the same once you're in the field. Many of these are easy to tell apart when we have museum specimens that are well curated in museum collections. However, one of the things to consider is that many of these specimens, once you're in the field, are extremely difficult to tell apart. This has been one of the things that we've been working on with many colleagues and students in places like the Field Museum. This has allowed us to better understand what species we even have in particular places. If you look at some of these specimens, they're easily discernible by things like characteristics and things like cranial dental items, characters. However, one of the things to keep in mind is once you're in the field, these characteristics are not available to you, and still identifying these specimens in the field is extremely difficult. As part of this, we have been working hard with museum collections to better understand the specimens, and this is important as we understand not only the number of species that we have in any particular place, but also the abundance of species, abundance per individuals at any particular place, which is another important and valuable characteristics of biodiversity. Now, in biodiversity research, it's important that we understand how biodiversity changes in gradients, for example, with fragmentation. Now, for many small mammal species on the planet, many of the traits that we use to understand functional diversity, for example, is based on categorical variables that really do not allow us to understand fine resolution trait variation, but rather end up with coarse resolution trait variation. And so therefore, it's important that if we are going to understand the impact of things like deforestation, that we have fine resolution trait variation. Now, these are important, and this is where things like museum collections are extremely valuable, as these allow us to look at really fine grain resolution with both categorical traits, but also with things like linear measurements, and also furthermore with things like CT scanning. One of the things that we were looking at recently was this analysis where we looked at variation in functional diversity, and we were able to look at things like locomotion, diet, and bioregion affinities. And this was based on a lot of research that was based on research from many collections that have already been developed. And therefore, we were able to use museum collections to improve on more quantitative traits that usually are not found in the literature, which hopefully will be improved with much of the work like the work that Brian is conducting. On top of that, like some of the stuff that Charlene was talking about, we were able to look at museum specimens to actually quantify things like bite force, in this case for rodents. As part of a study where I was able to compare past research and variation along a gradient on edge effect, I was able to look at new museum collections and then expand on the traits that we used in that particular study. And one of the things that we were able to do was that one of the things that we found is that when we use quantitative traits, in this case, all of traits that are right now housed in museum collections, we were to improve on the models that best explain functional diversity. Now, this is just one quick application of the many applications for museum collections, but as you can understand from the research that we've been looking at, there's a plethora of other functional traits that are just literally sitting in jars in many museum collections around the world. This is going to be in many ways the future of museum collections. One of the things that I would argue is that the future of museum collections is going to be in ecology and not so much necessarily in functional, not necessarily in evolution and taxonomy as has been used in the past. This is part of a study that we've been looking at where we're conducting a literature review of the applications of CT scans on mammals. You can see this has been increasing dramatically. And if you look at this, when you look at where ecology is in this spectrum, where it's not really included. However, you can see that morphology and evolution is one of the main sources of uses for these types of research. Rodents and many other small mammals, things like bats, are still widely used, but there is in some ways a disproportion of charismatic applications for rather the application of these types of research for charismatic megafauna. So as we look at this, the next trait or the next data sets that are extremely valuable are things like genetic data, things like whole genome sequencing, rat-seq population for population research, SNPs, various RNA-seq and Site-B-CO1. These are all data that should be associated with our museum collections if they're going to become more and more accessible for other types of researchers. Now, as an obvious example, one of the things that we are doing in the field, we are transitioning to doing things like mark recapture, which one would suggest is not usually part of museum collections. However, using the museum collections that we collected in the past, this has allowed us to better understand the species that we're capturing right now as we're doing mark recapture. This has been extremely valuable and has allowed us to go into the next phase of our ecological research that we're doing currently. So as part of that, this is the next part of biodiversity that becomes really important. And this is the application of phylogenetic diversity. This is extremely valuable as we better understand not only the functionality of the species that are found in a particular place, but also the diversity of those species that we're capturing. Okay, next thing that I want to get into is sort of the application for urban ecology, which is the next level that we're getting into. And this has become extremely important in Chicago, where we're trying to understand the population structure of rats in Chicago. Now, for the past few years, Chicago has been categorized as the rattiest city in the United States. I would argue that that is not correct. However, this seems to be the general trend in public media. Now, when we first started, one of the things that we wanted to look at was what is the population genomic structure of rats in the Chicagoland area? And we were trying to build on other research in places like New York City, where they've been able to do some pretty awesome population level genomic work. And one of the things that we wanted to do was we wanted to go back and look at all past collections for the past several, like 100 years and so. We're highly disappointed to find that in the Field Museum, for example, when we looked in there, we only found approximately 14 specimens for the area of the Chicagoland area historically. And obviously, since then, this was not what we wanted to work on. And so since then, we've actually started these collaborations with a local pest management company. And as part of that, we've actually increased our sampling considerably. And as part of that, we've actually been able to expand also on other specimens that were usually going to be destroyed without any real follow-up as part of pest control management. And now we're being able to take advantage of all that amazing biological infrastructure, or rather, we're able to take advantage of all that biological data that usually is lost. Again, when we start thinking about about museum collections, there is this this perception that we are affecting local populations, but there's so many other things that are truly affecting biodiversity throughout the planet that if we are able to take advantage of these types of collaborations and partnerships, we can gather a lot of information. One thing that was very prevalent and is probably prevalent in many museum collections is what we perceive to be the abundant, the common species, the generalists that we usually discard, are actually really poorly represented in museum collections. This was our example with rats in Chicago. And in many cases, many of the things that we perceive as abundant in our local environments are usually neglected. And this could be a wonderful opportunity to better understand things like, for example, urbanization, and especially with things like climate change in the next few years, ways that we are able to take advantage of collections that are usually lost. And so this is just generally speaking, just an overview of some of the stuff that we found. And as part of these collections, we've been able to understand population dynamics in the Chicagoland area. And also, we've been able to expand that to understand, based on social, economic, human covariates, what are the variables that best explain these genetic patterns. Right now, we've been able to show that things like sanitation districts and really the where and how trash is managed, those are the things that best explain our population structure of rats in the Chicagoland area. Now, moving forward and probably wrapping up here, the next part of this is how this is changed by landscapes and how this is going to affect our things like One Health. And as part of this is part of one of the things that I would like to sort of suggest is that the museum collections are just one tiny aspect of a much bigger ecosystem that has to incorporate things like the understanding of livestock and also how stress hormones are being influenced in different types of habitats, and looking at different types of tools, things like camera trapping, and then also things like bioacoustics and satellite imagery to get a bigger understanding of what's going on in different parts of the world. As part of this, we're looking at small mammal parasites. Right now, we're working on various things where we're trying to trace things like blood pathogens, looking directly at ectoparasites, but also at mammals, and trying to trace things like blood parasites. And more so, we're trying to expand into what's called VIRSCAN, which is basically, if you think about a multiplex ELISA test that allows us to screen for thousands of different types of viruses. One of the things that we're able to look at is all the different types of viruses that different types of organisms are exposed to. This is an example for one of our species. And moving forward, having access to this data associated with our museum collections are what's going to allow us to look at potential next zoonotic outbreaks in many parts of the world. These are just some of our preliminary data. I'm happy to elaborate more on that later. But just wrapping up, I think that it's important to consider the value that museum collections bring to larger ecological ideas, theses that seem to be unrelated to museum collections. And I think that bridging that gap between museum collections and field biology are the future of where museum collections are going. So I'll wrap that up there.
All right. Thank you, Noe. Could you stop sharing here? There we go. If everybody can turn their cameras on, that'd be great. So yeah, folks, if you can drop your questions in, I guess Shyla, I don't know how you if you want to take over now and
Sounds great.
okay.
Yeah. Thank you to all our presenters. That was an amazing webinar. If anybody has any questions, now's the time to drop them in the Q&A. I don't see any questions yet, so I will give it a second.
Yeah, I'll I guess ask a question just to kind of get it rolling. So I think thinking about the timeline of mammal digitization, right? And Nate, you said that you pointed out that it started 15 or not 1500 years ago, 500 years ago, roughly, right? The beginning of really sort of the natural history collections and their development. But for mammalogy in particular, and probably more broadly, the vertebrate collections, our databasing efforts, right, have been done for several decades. And now we're moving into this extended specimen concept with CT data, trait data, interconnections with web tools like Mammal Diversity Web and our database and others. What is next? I mean, I think Noe kind of touched on like some of the things we could be doing if we had other things available, plus like taking what we have right now and expanding on it. But where's the next sort of frontier in digitization for mammalogy? Brian, looks like you're unmuted, so I'll let you go first.
Well, I mean, we need to, there's little bits of data attached to specimens, right? And ranges is an example of going back and getting, predominantly the basic traits that underpin kind of our knowledge of body size and body shape. But there's other just bits of information we need to kind of build standardized ontologies for. All the other traits and genetic data that people extract from specimens. I feel like, if to extend the network, to extend the mammal specimen network, we need to kind of be able to track all those things and have clear, pathways and ontologies for how data get extracted, how sequences get extracted, who did it. It's very hard. I know that everyone on this call is familiar with this. It's hard to go back and build synthetic knowledge from all of these kind of pebbles and threads that take forever to kind of track down. So yeah, I would love to see like some kind of national infrastructure to really think about extended specimen networks and how to build them. And, mammals would play a big part in that, but obviously other taxa as well.
Other thoughts?
This isn't digitization per se, but I do see that a pivot in just preparation types. I think for so long the standard was a study skin, less so skeleton. But I think, we're realizing with CT scanning and of course other technical trends will come and go, but discarding a lot of that soft tissue anatomy, is not a great, I think there's so much utility into the whole organism and lots of new entry points for different types of researchers. So, we have parasitologists now that are utilizing collections that maybe aren't even interested in that mammal host, right? So if you're just keeping the entire anatomy, I think collections will just be ultimately higher utility. So I see a trend towards fluid preservation more and more.
Yeah, I guess I would say that, like, in terms of the next frontier, I mean, I feel like we're still like trying to figure out the current frontier. It's even things like georeferencing. I mean, I don't want to pick on the Smithsonian and American Museum, but like it's, most of the collection is still not georeferenced for a group like mammals. And that's, those are like the really foundational collections that are critical for baseline comparisons in modern contexts. And so I think we just need to like have a little bit of a reckoning in that context and really do more thorough gap analyses and make sure that the resources are going to filling in our top priorities, like as a in a global sense of where the data gaps are. And that just if we can do that, then we can actually start to show value for the digitization, and then the next waves can continue.
Brian, why don't you go ahead, and we got a couple questions in the Q&A we'll address next.
A quick comment that AI will be used and is being used to kind of analyze collections data, and we really have no framework in mammals or otherwise, as far as I know, to kind of make sure that those agents attach provenance and identity to any specimen data they use. And so I feel like this is one place where the mammal community could help other communities and lead, because we do have so much data digitized already. Are we perfect? No, but you've seen that like a lot of collections have their full kind of catalogs digitized. And so like, I feel like there's a chance to guide how the digital data are structured and make sure that AI agents can kind of use them in a sensible way, in ways that advance knowledge. At least we can kind of help them use, the data in a more trackable way.
So kind of pulling on into this conversation, so Lisa Walsh asked a question about soft tissue. What percentage of mammal collections currently have the space to expand their fluid collections? Emily, I can speak to my collection, but I'll let Emily answer as well, and maybe others.
I'll say, I mean, space is a limiting factor, I think, for all of us. But, in some ways, it is a more compact way of storing multiple specimens together. You're essentially, I mean, I don't know about everyone, but you can put multiple specimens in the same jar or the same tank because they are individually tagged. So essentially making it almost lot-based. And, you are saving on other things in terms of prepping out a full study skin or skeleton, so that there's a time-saving element to it as well. But yes, space is certainly a concern.
Charlene?
Yeah, I think Emily basically captured the two things that I was going to say in terms of saving time, and then the amount of data that can be extracted from the fluid specimen is so much more that it is, even if it did occupy more space or had other sort of repercussions in terms of specimen storage, I think it's really worthwhile. I can speak, for our collection that we do have plenty of space to expand mammal collection as long as they're small mammals. The challenge becomes when we're talking about small mammals versus large mammals, and things become, of course, more complicated. Maybe some parts of interest of the larger mammals can still be preserved. But definitely, I do agree with the things that Emily said.
Okay, and Charlie, you might want to stay unmuted. This would seem like this would be a good question for you from John Woodward. On the topic of 3D imaging, I wonder if anyone here is using Gaussian splatting either for documentation and distribution of specimens or for crunchier or more researcher-y applications.
Yeah, I did see that question. I wish I knew what Gaussian splatting is, but I have never used that technique. I am guessing that maybe it's some computer learning algorithm or some AI related. Some clarification would be useful. If that's what it is, I think the question, let me just go back, because the question was about specimen documentation and distribution of specimens. I'm also not entirely sure what that means. For researchy applications, there are people who are developing tools for computer vision, where you can train an algorithm to segment out specific features of a scan, for example. And those so far can work fairly well if you are working within one species and you're isolating one or a set of structures. There's still limitations when you are comparing species that have very different morphologies, and that's where the algorithm fails. I think there's a note. Let's see, there's a note in the chat here. Model 3D scenes, cells are optimized. Okay, yeah. So training, so similar of the type of methods that I'm thinking about in terms of training an algorithm to segment out. Yeah, so there's still, yes, that's definitely something that people are thinking about, and there are some advances in development, but we're still, I would say, it's still at its infancy, and that's what I said in my talk, that still the human eye is the best tool that we have for segmenting these morphologies, especially if you're working with very disparate types of morphologies. But yeah, I'm sure we'll get to some automation very, very soon, at least for some of these tasks that take a lot of time at the moment.
Well, I don't know if we have any more time for questions or not, or if Shiloh or Gil or any of the iDigBio folks want to close us out, or.
Well, I see Gil's unmuted. Yeah, sorry.
I have a question, Cody. It sort of relates to what Brian was talking about, but I think it relates to a bigger issue than that. I'm not even totally sure how to ask it, but I mean, if you look at the people in this room, this is a highly specialized knowledge that's very important for the future of Earth. No question about it. There's a lot of passion and enthusiasm in this group. How do we help the rest of the world, particularly the rest of the general public, gain some of this knowledge so that they are more passionate about and understand better why it's important to preserve and conserve what our natural environments have? And maybe Brian's right. Maybe that's one of one of the services that AI could provide is through bots and stuff, helping people understand better how this stuff works and build some passion. I just
Yeah,
think that's what we're always talking about,
yeah.
wanting to do.
I know I've had definitely with Noe, I've had different conversations. I thought certainly I think the for use at the university setting, we need to get back to teaching specimen-based courses, right? We need to get students seeing things in classes, and going out to the field and things like that, right? We've kind of gotten away from that in recently. And so reinvesting that would be is much needed. The nice thing about mammals, right, is we're mammals, right? I think that's the other thing for mammalogy is that we're people like going to the zoo and seeing elephants and paca and things like that, right? Those are fun and bats when you look at their facial structures and some of the they're beautiful, right? So for us to sort of build into that and that's it's basically shovel ready to me for general public. And if you digitize image things, we can really get these collections in front of people in a more larger effort, I believe. So, Nate, or
Yeah, no, it's a really good question, Gil. I guess I have the privilege of teaching a large enrollment evolution class, and so there's like we have like 300 students, and I get to talk to them about the biodiversity crisis and potential solutions. And part of what we end up talking about is that a lot of people have the sense that they're sort of alien in nature, and that like we are outside of nature, and so therefore we have the power to like dominate it and control it and things like that. And so a lot of what I try and emphasize is that like we grew out of the earth, like we didn't come here, like we're from the earth, like we're part of nature, and that a lot of these distinctions are because of our language rather than um, anything that's actually real. And so I think the more that we can show these ecological interactions, including at multiple levels, including microbial interactions in our cells, and just like all of these things are connected. And the more that ecologists can communicate that and do outreach to show the interconnectedness of things, I can only hope that people would then not want to destroy it, right?
Yeah, right. So I think you must be talking about a course like what FSU used to call baby biology, where you had several of the better professors that really could communicate, would teach the same course, and each one would do one piece of it.
Hmm.
And so they were able to really highlight their area, but they did it in a way that was enthusiastic and engendered interest. And I think you're right, Cody, that that's the kind of stuff we have to do more of and get people really. I think one of the most important things my child did when she was a little girl was go to this Saturday at the Sea, which was one of the guys in biology at FSU would take a whole group of kids down. They were 4, 5, 6, 7, 8 years old, and have them dig around, play around in the water and dig around stuff and find things. And so to me, that's, you've got to have that fascination first. And then after that, we may have be better off. Anyway, I'm sorry, Brian, let me to break in.
Brian, I think Noe wants to say has something to say after you're done.
Yeah, real quick. I mean, I think I'm really excited to be in this panel because this is like Gil said, this is people who are really kind of passionate about mammals and extending what we can do with collections. But I also know all these people, and I know all these people are really passionate about being out in the field. And I take students out in the field as much as I can, and that's a great way to make people feel like they're not alien. And it's not new, right? This is what, I mean, going back to Cody's talk at the very beginning days of all the taxa at collections, right? People were going out in the field and like we don't need to trip ourselves out, right? That's what we still need to be doing, at least in part, to get people to understand nature is we need to be doing this basic stuff that's been done for the last 150 years of collections. And I try to do it as much as I can. Is it easy? No, it's like a lot more work than giving another lecture, but it kind of pays a lot more dividends too,
Yeah, I think you're right.
Yeah.
And I think going out in the field with people that know stuff
Mm-hmm.
is super helpful for young people who, we used to do a lot of field trips with plants and some natural areas, and the kids were always amazed. They would say, "Geez, do you know
Yeah.
every plant out here?" And I
Yeah.
no, working on it. But that's what I think, that passion for knowledge is important. No, you had something there.
Yeah, so to piggyback on everything that's been already said, I think that moving forward, an important thing is that, especially with AI and stuff, we'll be able to go get back to the original questions that were of interest to us, right? Being able to go back to questions dealing with mammalogy, dealing with being out in the field. As we all probably all know, academia in general is becoming in some ways prohibitive for many people, especially the next generation of students. Getting a faculty job is not the same that it used to be, especially if we want to focus on these types of jobs. As we've been going more into more sophisticated techniques, where even people who are biologists, instead of being mammalogists, they're computational biologists. They're genomicists, which is great. But hopefully, when these become more and more automated, is the tool is not the main driver of the science, but rather going back to the original questions that are of interest of mammalogy, right? So hopefully we can have mammalogists who can have a career as as a professor, as a mammalogist alone, not having to be a computational biologist, not having to be some kind of other highly tool specific specialist, right? And that's what I'm hoping that things like AI and stuff will allow us to do, is go back to the original questions that are of interest to us and have a situation where in the United States we can have professors who can live off of just being a mammologist. And I think hopefully this is something that we can keep moving on this.
Good point.
Thank you so much. And Cody, if you need to go, thank you so much for joining. And I just want to extend a sincere thank you to all of our presenters. Before we conclude, I wanted to remind everybody that, like with all other iDigBio webinars, this has been recorded and will be posted to our updated YouTube channel. It is now available, and there you can find recordings from this webinar and recordings from past webinars in the past, as well as other iDigBio events. And our next webinar is on May 5th and is titled Imaging the Florida Museum's Natural History Collections. Registration is now open, so we encourage you to sign up. Another friendly reminder that the Digital Data Conference is happening virtually from June 2nd through the 4th. We're hoping this won't be the last conference, but it is the last year it is free. So definitely take advantage while you can. The deadlines for abstracts is closing very soon on May 1st. And go check out the conference page for additional information about speakers, themes, and accessibility. We hope to see you there. And finally, follow us on social media to stay up to date. And again, thank you so much to all of our presenters.
I would like to echo Shiloh's thanks. You guys, this has been really fantastic. I mean, it's just flown by, and the work you're doing is really phenomenal. And I have certainly appreciated hearing all about it.
Thank you guys so much. And I'll go ahead and close things out.
Thank you. Bye.